slides for you. And during the question and answer, virtual participants, please use raise hand or chat function in Google Meet. And in-person participants, we can just raise our hand. And we have lunch at 12 p.m. It's on your own. I guess we can walk to the cafeteria together if you like. And we have 2 breaks, one at 10:20 a.m. and the other in the afternoon at 2:20 p.m. That's about what I would like to see. And I will stop sharing. And our first speaker, let me check the schedule, is from Diptimi Behera. Sorry if I say your name wrong. And could you please try to share your screen? Thank you.
Hello everyone. I hope my screen is visible. And okay, I guess I'll get started with my presentation. Please let me know if I'm not audible or if my presentation is not visible.
We can see your slides, but it's not in full screen mode. You please quiet. Okay, it's better now. Thanks.
Okay, thanks. So, hello everyone. My name is Diptimi Behera, and I am a PhD scholar at the Indian Institute of Science Education and Research, Mohali, in India. Most of my PhD work is based on delineating natural and anthropogenic sources of sedimentary organic matter in various aquatic systems. But I have got the opportunity to work on several collaborative projects focusing on paleo-environmental reconstruction over different timescales, such as Quaternary, Myo-Pliocene, Eocene, and even Permian Triassic. And today I shall be presenting on one such work in which we have reconstructed the past climate and vegetation using molecular approach on lignite samples from southern India of the tertiary time period. So, introducing the positioning of the continental plates is not static even throughout the geological time scale. Along with the tilting of the earth and the positioning distance from the sun, the positioning of each continental plate is crucial in determining their respective climate and vegetation profile. So if we look,
have you moved on to the next slide? Because we're still seeing your title slide. Oh, okay.
Okay, there we go.
Yeah, it's good.
We are at the second slide now.
Okay. So the positioning of the continental plate is not very static, and it keeps on changing over the geological time scale. And these all this is also a crucial factor in their climatic and vegetation pattern. And if we look at the Indian subcontinent in particular, it has not been the similar way it is now in the present scenario. Initially, during the Paleogene time period, it was far away from the equator. During the Eocene, it moved further towards the Eurasian plate, while positioning itself at the equator. After that, during the middle to late Eocene, it collided with the Eurasian plate, moving further from the equator. And finally, during the Miocene, it established itself with the Eurasian plate, thereby forming a full-fledged orogeny of the Himalayan mountains. And this brings us to the present scenario where it is very far away from the equator, lying in the subtropical and tropical zone, thereby having a climate and vegetation of the tropical and subtropical zone. So there are two significant different positioning of the Indian subcontinent. One is during the middle, early to middle Eocene, when it was at the equator, and then when it was farther away from the equator. Therefore, reconstructing the paleo environment during the Eocene on the Indian subcontinent is essential for understanding the geological, climatic, and evolutionary processes that have shaped the region over millions of years. This knowledge not only contributes to our understanding of the Earth's history, but also has some practical implication for environmental management, as during the Eocene, the temperature was very high than average, which is very much similar to the present scenario where we are facing a concern of global warming. So, as you can see, a couple of works has been carried out in understanding the paleovegetation and climate of the Indian subcontinent. For instance, in the western part, Sukla and Mehrotra 2018 constructed that during the Eocene, the climate was very hot and humid with a tropical rainforest kind of environment. Similarly, Prasad et al. 2018 constructed the past climate to be tropical rainforest in the northeastern part of the subcontinent, whereas the northwestern part of the subcontinent was mostly dominated by mangrove forest, as detected by Prasad et al. 2019. However, a contradicting study is also present from the same region, stating that although the climate was hot and humid during the time frame, but the vegetation was mostly conifer mixed tropical forest, as stated by Chieta et al. 2022. However, there are not many exclusive or extensive studies carried out in the southernmost part of the subcontinent during the Eocene period, although sections are available to this particular time frame, which brings us to our objective of this work. For this, we selected the Neyveli lignite deposits present in the southernmost part of the subcontinent in the state of Tamil Nadu. The chronology of this particular lignite deposit has been established to be from late Paleocene to middle Eocene, thereby contributing to our investigation of paleo environmental construction. So, to proceed forward with our objective, we need to apply a proxy. So, as we know, there are many kinds of proxies available to address our objectives. There are diatoms, there are pollen, there are geochemical analysis, but depending on our on the chronological constraint and most importantly the available technology, we decide the proxy which we can use for our analysis or reconstruction of paleo environment. And in our case, we use the biomarkers or the molecular proxies to reconstruct the paleo environment during this particular time period. So the very first question arises is, what are biomarkers? Just as the fossil of an organism can be used to determine the presence of that particular organism, similarly, there are certain molecular fossils which can be used to determine the presence of a parent molecule or the parent organism or a particular environment that produce that particular molecule in a given frame of time. Such fossil molecules are termed as biomarkers, and they can be used to trace back the parent organism that produced them in the first place. Now that the proxy is clear, I would like to discuss further how we can integrate it into our understanding of the sources. Now, what happens in an aquatic system in particular is that organic matter is derived from various sources which are categorized into either allochthonous or autochthonous sources. Allochthonous sources are the ones which come out of the system, such as terrestrial input, whereas autochthonous sources are those which are produced within the system, such as macrophytes and microbial communities. Now, these dissolved organic matter is hydrophobic in nature, and that's why it doesn't have a very high residence time in the water. It tends to absorb onto the surface of the sedimentary matter and remains preserved unless and until some amount of diagenesis or degradation occurs onto it. And hence, if we do the molecular analysis or the biomarker analysis of this sedimentary organic matter, we can trace down the sources and categorize them into allochthonous and autochthonous sources. And eventually, from this directly, we can determine the environment as well as the climate in which these particular sources could have been prevalent. So with this knowledge, we moved on to our protocol, which was established for the biomarker analysis. So the very first thing we need to do is remove any amount of moisture present in our sediments or lignite samples. For that, we either use freeze dryer or hot air oven. After that, we homogenize our samples by mortar and pestle. After that comes the extraction part, in which our target is to obtain the total lipid extract. This is the total lipid extract is a mixture of various molecules which were initially the building blocks of living organisms and are now used as biomarkers. So for the lipid extraction, there are many protocols or methods available, such as sonication or Soxhlet extraction or accelerated speed extractor, and various combination of organic solvents are used to obtain the lipid extract. In our case, we use the accelerated solvent extractor with the organic solution combination of DCM and methanol 93:7. Now, this combination and the method which we use for the extraction can vary depending on the nature of the sample which is being investigated. If the organic content is very high, such as the case of plants or coal samples, we can use sonicator or Soxhlet extractor. However, if the organic content is very low and you have to use a lot amount of sample, we go for the accelerated solvent extractor, which makes our work easier. Now that the total lipid extract is obtained, as I mentioned earlier, it is a mixture. It's a mixture of vast number of compounds, and if we analyze it as it is in the GCMS, we cannot separate it or analyze any compound. So to make the analysis more clear and viable for us, we do the liquid chromatography or the separation of the compounds into various hydrocarbon fraction. Typically we fractionate it into three parts. One is the saturated hydrocarbon fraction, the other is aromatic hydrocarbon fraction, and the third one is polar hydrocarbon fraction. And after that, we analyze it in the GCMS and interpret each hydrocarbon fraction based on our objectives of the study. And for other than that, we also maintain quality control to make sure that we extract we recover the maximum from this protocol. This begins during the pre-process time, where we bake all our glassware at a temperature above 300 to a temperature ranging to 800 to 1200 degrees Celsius for a time period of 8 to 12 hours, to make sure that there is no moisture or organic content in them to begin with. We also make sure that none of our samples are exposed to any temperature above 40 degrees Celsius. For that, we maintain a temperature of 35 degrees or 30 degrees in a hot air oven, or we use the process of freeze drying to dry our samples. When it comes to the sample analysis, we make sure that the instrument is functioning at its very best. For that, we regularly auto-tune the instrument, followed by cleaning of the auto sampler. We also make sure that the column is also functioning at its best by cutting the column or replacing it if it is too brittle. We also run frequent blanks between each sample, thereby assuring that yes, the instrument is working at its best. And last comes the post data acquisition QC. What we do is add a certain deuterated form of the expected compounds to the samples at the beginning of the process. So when we carry out the GC analysis, we can account for any kind of human error or loss of sample based on these deuterated compounds. And we have a average recovery rate of 86 to 98%, which means that the protocol is indeed reliable, and there is not much error associated with our protocol. So once this is done, we move on to the results part. So for this study, we just analyze the saturated hydrocarbon fraction, and here is a representative chromatograph. What we each peak in this chromatograph represents an organic compound, and in this case, since it is the saturated fraction, it represents mostly alkanes and isoprenoids. And the results which we have obtained includes N-alkanes ranging from C16 to C35, and certain isoprenoids which are termed as pristine and phytane. Now the question is, what is the implication of these compounds? So I'll start with explaining the alkanes first.
Sorry for interruption. Could you please
Okay.
wrap up in a couple minutes? Thank you.
Yeah, sure. Okay, so N-alkanes are produced by various groups of organisms, such as the short chain N-alkanes are produced by microorganisms, whereas the mid-chain N-alkanes are produced by the aquatic plants, and the long-chain N-alkanes are produced by the terrestrial plants. This has been proven by various studies, and I have also proven it in one of my study, in which we took fresh living plants and algal mat to determine or cross-check these results, as you can see. But the problem with this interpretation is that it is a spectrum of data, and we do not have a quantified value to determine the source of the organic matter. So for that, there are certain N-alkane indices which has been developed to quantitatively determine the source or the predominant source of the organic matter. So one such index which is being used in the study is CPI, where the values less than 1 indicates degraded or diagenetic product, or and the value above 1 is indicative of fresh organic product, and our values mostly lies above 1, indicating fresh organic product. Similarly, ACL is used to determine the contribution of terrestrial sources to the organic matter, and our values is very high, more than 28, indicating high contribution of terrestrial matter to the organic source. Then further, we did the TAR index, which indicates the contribution of terrestrial sources with respect to microbial sources, and most of our values showed high terrestrial contribution and low microbial contribution. We also did the P-aqueous analysis, which is used to determine floating, submerged, or emergent macrophytes in contribution to terrestrial sources. We found that most of the organic matter is indeed contributed by emergent macrophytes or terrestrial sources. Finally, we moved on to the isoprenoid ratio, which is used to determine the redox condition of the environment. Most of the pristine by phytane ratio lied below 0.8, indicating an anoxic environment. However, certain fluctuations were seen throughout the trend, which indicated some amount of suboxic condition might have prevailed. Other than that, we also, well, we are talking about molecular or chemical signatures of the compounds here. There are particular compounds which can be used to determine the source, such as these triterpenoids, which are direct indicators of presence of angiosperms. And there are other indicators of gymnosperms, which we did not find in this study, thereby determining that the major contributor were angiosperms in this study area. Other than that, we found very low microbial contribution, but with certain compounds, we can establish that the microbial contribution was significant, such as these tetraterpenoids or hopins, which are formed from the cell membrane of certain bacteria and other microorganisms. So in conclusion, I would like to state that the environment was quite arid, as you can see from the high ACL values. Also from the P aqueous values, we have determined there was a dominance of emergent macrophytes in the system. There was a slight amount of fluctuation in the water level as indicated by the pristine phytin values. However, this cannot be further confirmed. This has been further confirmed by using several other proxies such as compound specific analysis and petrography, which I couldn't discuss within this given time frame. However, this is what we have concluded about the paleo environment and paleo vegetation of this particular region. And with that, I would like to conclude. Thank you very much for giving me this opportunity. And any questions?
Thank you for the nice presentation.
Okay.
Do we have questions? Okay, thank you again. And in the interest of time, we can move on to the next presentation. It's from Rich Fiorella. Rich, are you there?
Yep, I'm here, John. So,
okay, please share your slide.
Sure. Okay, how about now?
That looks good.
Great, thanks. Yeah, so everyone, Rich Fiorella. I'm currently at my primary affiliations at Los Alamos. I'm presenting work that I've done kind of on an NSF project through the New Mexico Consortium that won't strictly be paleoclimate today, but we'll introduce a tool that I think will be of pretty broad interest and utility to paleoclimate research. And so I'm going to focus mostly on water isotope ratios and the ways that we can understand how they vary using Earth system models. So water isotope ratios have drawn a lot of interest for paleoenvironmental reconstructions for a number of decades, since they have a strong spatial pattern is directly related to climate. To a rough approximation, we can explain the distribution of modern precipitation isotope ratios is varying with temperature. There's a bit of a different structure in the tropics. But generally, isotope ratios decrease in precipitation at high latitudes and altitudes and far away from oceans in continental interiors. And in addition to their relationship to the direct relationship to climate, water isotope ratios are attractive for paleoenvironmental reconstruction, specifically because water is either preserved directly in the case of ice cores or proportionally to environmental water. And materials such as tree rings, soil carbonates, fossil teeth. And so there's a lot of proxy materials that are based on this isotope systematics. And just to give a quick primer on isotope fractionation and the kind of the processes that are responsible for the spatial distribution. These spatial patterns and water isotope ratios ultimately arise from an unequal partitioning of isotopes of oxygen and hydrogen during phase changes that get mapped onto atmospheric water transport. And so oxygen has 3 stable isotopes, 2 of which are more commonly used in studies, oxygen 16 and oxygen 18. Hydrogen has 2 stable isotopes, so hydrogen 1 and hydrogen 2 or deuterium. And this produces water molecules that have different masses. And this difference in mass drives the unequal partitioning of these isotopes during phase changes, where during condensation, the heavy molecules preferentially condense into the liquid phase, while during evaporation, it's the opposite. Lighter molecules preferentially evaporate. There's a few pieces of standard notation about isotope ratios that I should introduce because they're used throughout the rest of the talk. First, as I mentioned a lot of times, these tend to be isotope ratios expressed as R, which in the case of water isotopes is the heavy to light isotope ratio. There's another parameter called alpha, which describes the degree to which these heavy and light isotopes are partitioned across phases. And it's a ratio of these isotope ratios. And so here it's usually defined to be greater than 1, because we could expect the heavy isotopes in the liquid to be more prevalent than in the vapor. We expect this ratio to be higher than this ratio. And finally, as these variations are small, We tend to express isotope ratios and delta values as part per thousand deviations from a standard isotope ratio. And for water isotopes, the standard is a mean of ocean water. The most commonly used theoretical framework to explain variations in precipitation isotope ratios is Rayleigh distillation. And this describes how water vapor and condensate forming from water vapor in an air mass would evolve as water is continually removed in an open system, single source condensation. The figure right shows how vapor and liquid isotope ratios would co-evolve as water is removed. The initial fractionation here for oxygen is about 10 per mil. And both vapor and liquid isotope ratios decrease as heavy isotope logs are removed from the system. The Rayleigh equations describing this relationship are shown on the bottom left. I put them in both isotope ratio notation and R on the top, and delta notation on the bottom. And we can observe from these equations that there are three factors that can vary that give rise to our isotope distributions. There's the initial isotope ratio, delta 0. There's this fraction of vapor remaining, which I'm showing here in terms of specific humidity is Q over Q0. And then there's alpha, which describes the partitioning between the phases, how strongly it's partitioned into the liquid. The heavy isotopes are partitioned into liquid. But because F is the exponential, is an exponential term here, and you can see in the shape of the curves, it can have changes in F or Q over Q0 can have a really big impact on delta. And so delta is kind of most naturally interpreted as a measure of rainout or changes in F. And so this Rayleigh distillation model has been used for several decades at this point, basically since the 50s. And it's remained popular because to a first extent, even though it's a pretty simple model, it works pretty well, at least at large spatial and temporal scales. Though it was known to have a few limitations fairly early, and as more data have been collected, there have been more situations where the Rayleigh model doesn't appear to match the observations. So for example, in these papers, these authors talk about changes in kind of source compositions and changes in upstream precipitation processes, or changes, seasonal changes in where the vapor is coming from, or perhaps these isotopes vary with atmospheric residence time of that water, or the type of cloud that they're formed in, or the kind of vertical profile of moisture entrainment into the cloud, large-scale patterns of evaporation, precipitation, or some authors have just kind of concluded all of these factors play some role, and so we need to be aware of them. And so one of the things I've been thinking a lot about in the past few years is how we can use our system models to investigate how isotope ratios depend on these various processes, and how we can use them, use isotope ratios in turn as constraints on model parameterizations and processes. And this first figure I'm showing, this is results from a run using isotopes from CAM6, using an FCOM set. It's about 1 degree nominal resolution. And the winds have been nudged to the ERA5 reanalysis. And then the contour plot shows weighted annual mean precipitation isotope ratios, which we generally see the expectation following the expectation that we had from the observations, where we have high delta values in the tropics and lower values at high latitudes, altitudes, and in continental interiors. The figure on the right shows a comparison between CAM6 and CAM5 against observations. And on a long-term basis, though there's some spread around the relationship, CAM6 shows a little bit of improvement over CAM5, but both models are able to fairly well capture the observed range of precipitation isotope ratios. That's great, but we're often interested in more than the isotope ratio itself. And often the isotope ratio, when we go to measure it, particularly in a paleo context, stands as a proxy or some measure of a process or a variable that we're more interested in constructing. And so I've been working on extending the water isotope framework to allow tracing of specific processes associated with evaporation, condensation, and transport throughout the model. And so in each of these panels, the value at each cell shows some property of precipitation in that cell, but it may be integrated kind of over all of the moisture that contributes to precipitation in that cell, which could be in different cells and different times. So in the top row, I'm showing an example of tracking a particular flux. On the left, we're looking at the amount of precipitation at that location that arises from transpiration in that cell or elsewhere. On the right, we're looking at a condensation property. In this case, the temperature at which water in that precipitation condensed, which is useful both for understanding isotope ratios, but also for understanding energy balance and transport. In the bottom row on the left, showing the mean relative humidity during evaporation experienced by the water in precipitation at their location. And then on the right, this is kind of a partial integrated property where I'm showing the mean time that water vapor that ultimately forms precipitation in that location spent in the atmosphere between source and sink. And so now to kind of lay out specifically why these might be useful for a useful tool for helping with analysis of paleoclimate data and model simulations. I'm going to spend the rest of the talk kind of looking into what these tracers can actually tell us about the drivers of precipitation isotope ratios. And so looking at 2 points. First, I mentioned that there's been a lot of discussion in the literature about apparent non-Rayleigh behavior. So we'll reevaluate how well the Rayleigh distillation model works to describe precipitation isotope ratios. And then second, a second order isotope parameter, deuterium excess or DXS, which relates the hydrogen isotope composition and the oxygen isotope composition, is often thought to preserve information about the evaporative source, such as its temperature, relative humidity. That assumption has been used to in kind of reconstruction of past climate and ice cores. And so we'll take a look at what information is actually recorded in DXS using this tracer framework. Okay, so first looking at Rayleigh distillation and how well it can explain isotope ratios. Give a quick walk through these plots. So we're going to be showing the delta 18O from the full physics in CAM6 on the y-axis against a derived quantity of how much of the vapor is remaining in the parcel, so the ln of f. I'm showing this in log notation as shown by the equation at the bottom here so that I can describe delta as directly proportional to ln of f. And so this red line shows a theoretical expectation for isotope ratios that follow Rayleigh distillation if alpha were constant at 1.01 and starting at an initial isotope composition of 0 per mol. The right panel differs from the left only in that I've restructured delta from the full physics here to actually describe, to remove any variation from the initial isotope ratio during evaporation that comes from different environmental conditions. So if we now look at the values from CAM, we see that they generally tend to fall along this theoretical line, particularly at high ln of f. But there's some spread around here, and then they start to diverge along a different line at high rain out or low ln of f, where they end up being lower than what we'd expect. If we remove the variability from the initial source conditions, I see that a lot of the spread at high ln of f goes away, indicating that a lot of this variability has to do with those variations at the evaporative source. And then the deviation at low ln of f, don't have time to get into this, but it's due to ice supersaturation and how alpha changes at low temperatures. What about DXS? So we look at DXS when information is contained about the evaporative source. I'm now showing plots that look at the mean evaporation, RH, temperature, and wind speed corresponding to each evaporative driver of kinetic fractionation that comes from the Craig-Gordon model for evaporation. And so the left column actually shows the correlation between evaporation relative humidity and precipitation DXS at that location. Um, and the right column shows the strength of that relationship. So basically, how much of a change in some remote evaporation quantity would affect local precipitation d-excess. And so for relative humidity, we see that there's a strong negative relationship between relative humidity and d-excess, with lower relative humidity implying higher d-excess over much of the globe, with perhaps the strongest signal in East Antarctica here. For temperature, we see a negative negative correlation in the extratropics primarily, but a positive correlation across the tropical oceans. And so the magnitude of the temperature effect appears to be strongest over the tropical oceans as well, where it can exceed about 1 mil per degree. And finally, for wind speed, we see a negative correlation and a large magnitude signal in the tropics, but a positive correlation in the extratropics. One last thing that makes these really powerful is that we can now fit different types of models to help unravel some of the controls in an integrated and quantitative way that describe the variations in isotope ratios. And so here I'm showing results from a random forest regression model on time series of precipitation isotope ratios and various drivers such as the rainout fraction, condensation temperature, evaporative conditions for d-excess here on the right. And I guess one important point to make here is that none of these variables are available from the standard model output. They all originate from the tracer code, and this allows us to connect model variables to the actual underlying physics that we think describe the situation from theory rather than, say, using local temperature, because this is what we have available. The x-axis is a normalized variable importance metric. Essentially, it's how much worse does the model do if we leave that variable out. And running this model globally, we find, so for delta 18O, the most important variables are this rainout fraction, and then the temperature at which the precipitation is formed is second. And for deuterium excess, rainout fraction plays a large role over land. This is largely driven by Antarctica. But particularly over the oceans, we see large roles of evaporative conditions, particularly over the ocean and for relative humidity. We can also look at the sensitivity of these parameters to changes in drivers using these random forest models, which allow us to
Rich, sorry for interruption. Please wrap up.
I have one more slide.
Yeah, so, thank you.
Yep. This allows us to look at expected isotope ratios across the range of values that these driver variables take. So in this example, I'm showing how d-excess would be expected to evolve with changing Ln of F in the top row, and then evaporation relative humidity in the bottom row. This can reveal some of the nonlinear responses of these variables. So, for example, if we look at relative humidity here, it's linear for a large portion of the range, but kind of saturates at high relative humidity and low relative humidity, indicating that other variables take a larger role, describing the variability of those. Now that I'm at DOE, primarily I've moved to working on E3SM, and I've shown examples of how these can be useful in a single model so far, just in the atmosphere. And I'm starting on a new project in E3SM where I'd like to build these tracers in a way that they can be passed between submodels. So the top figure shows kind of showing the bulk fluxes now, and the bottom figure is how we might be able to pass components of that bulk flux around and look at that. And the project will be looking at this capacity to look at interactions between urban and coastal systems with their environment. And one thing I'd like to highlight, I will be hiring a summer student intern at Los Alamos for this project this year. If you either have a student or are a student that might be interested in working on something like this. And with that, I'll wrap up to acknowledge a few other collaborators that have worked with me on this project in different ways, and then NSF, Climate and Large Scale Dynamics, for funding the CESM work, and the DOE Office of Science for funding the E3SM work. And thanks.
We have time for questions. Any question? Ryan, please go ahead.
Hi, Rich. This is very interesting. I have a quick question. How do you quantify the ring knot fraction? Because it's hard to wrap my head around when you have, because the model
Yeah.
obviously is Eulerian and it's not Lagrangian, and it's really hard to know like what do you mean by what is like a vapor source?
Right,
How
that's a
much
good-
is it?
That's a great question. So I will share my screen again. I have an extra slide that goes into that. Okay. Can you see this now, Ron?
Yeah, I can see
Okay,
it.
great. And so the way that I've done it is maybe a little bit different than the way that we think about the Rayleigh model typically, where you set a source. But essentially what I'm doing is if we fix alpha to be constant and then we fix delta 0 to be constant everywhere, we have kind of an idealized constant fractionation delta that then we can use, since this is fixed and this is fixed, right? We can just calculate ln of f directly from that. And so this f does take into account like mixing of air masses, right? And types of things. So it's maybe a little bit differently than the way that we tend to think about it as a single source. But this accounts for kind of all the different sources. Does that make sense?
Okay. Yeah, this question makes sense. So the delta 0 would be different. Let's see if you go to a paleoclimate state, right? If you want to make similar calculation.
It doesn't need to be. It could just be like there's nothing magical, I guess, about where you fix the delta 0 to be, right? I just chose 0 because it's convenient in this case. But yeah.
Okay. Okay, great. Thank you.
Yep.
Another question, Rich.
Yeah.
So you showed the comparison of model results against present day observation of delta 18O in precipitation.
Mm-hmm.
You didn't show the deuterium excess. You did mention a lot of the processes. I'm wondering how does that compare with observation? And I know there are biases there.
There are,
Do you
yeah.
have any insights about how to improve the modeling of deuterium excess? Thank you.
Yeah, that's a good question too. So so I guess these are pretty close to the 1-to-1 line for delta 18O. For deuterium excess, it's actually pretty much essentially uncorrelated in CAM 5 and then more weakly correlated in CAM 6. So it's a little bit better in CAM 6. And my hunch about what is there are two things that I think are driving problems with the d-excess. So first, it kind of low delta values. I think it is probably related to how we treat supersaturation over ice in CAM 6. So say for Antarctica, it's really sensitive to changes in those parameters, and I'm not sure that we've fully optimized that yet. At higher delta values, I would suspect it's probably most likely related to how we treat subcloud evaporation of raindrops. And there are some things that we could do to improve that. But part of it also could be related to the initial deuterium excess of the evaporation, and maybe we're not capturing the evaporation process perfectly.
Okay, thank you. This is helpful.
Yep.
And we move on to our next talk, Sophia.
Should I share right here?
Yeah, you can use it. I downloaded your.
Oh, you got it. Oh, great.
Yeah.
Okay.
So we can use our own computers, will still show up on Google Meet, okay?
Yeah.
Yes.
Just making sure.
Should I sit here
This afternoon.
Yeah.
or should I stand up?
See how it works for everyone else first.
Yeah,
Can people online hear me if I speak right here?
you have to speak loudly.
Yep.
They have us here.
Oh, you.
Speak loudly for us.
Or I can sit right here? Okay. Okay, perfect. Okay, yes. Question? Oh, no, just a hand raise. Okay, perfect. I'm Sophia Makarowicz. I'm a project scientist in the Paleo group at NCAR. And today I'm going to talk about evaluating water isotope tracers in the variable resolution community Earth system model. And although this is not an explicit paleo talk, I think this is a modeling configuration that could be really useful for paleo. And so I'll try to connect it back in the end. So the reason why we're interested in variable resolution approaches to isotope-enabled climate modeling is because variable resolution grids can capture both global climate dynamics and processes, as well as fine-scale details in regions of complex terrain at a relatively low cost. So here I'm just showing 3 different cost estimates from CESM2 runs, where you have a typical 1 degree low resolution run in the bottom left, and you can contrast that with a high resolution global run at 1/4 degree resolution. That's very high in cost. And then you have this CONUS run, which is a variable resolution grid that you can see in the bottom left part of the screen, where you have 1 degree resolution that gradually refines to 1/8 degree over the contiguous US. And so although there are a lot of benefits we could see from variable resolution modeling for understanding water isotope ratios, a configuration with water isotope ratios has not yet been tested. And so in order to test a variable resolution simulation, we need a data set that is dense and has continuous observations. And so the US network of isotopes and precipitation provides this with weekly observations from 1989 to 2014 of precipitation delta O18 and delta deuterium at 73 sites over the US. And so this is going to show us the spatial and temporal variability in precipitation isotopes across the US and help us validate this simulation. So I'm going to show you 2 different simulations with CESM2 today using the SE DICOR. So both of them have the community atmosphere model version 6 with isotope tracers in it, coupled to the community land model version 5. There's not fully represented isotopes in the land model, in this version of the land model, so we use a bucket model instead. And then also there's a prescribed ocean condition in both the simulations. So prescribed sea surface temperature, sea ice, and isotope ratios in the seawater. And so we've got a low resolution run with 100-kilometer resolution, and then also this high resolution CONUS run with that same, 1 degree refining down to 1/8 degree over the US. And these are both run over the same historical period as US NIP, and then they're both nudged with respect to temperature and winds to MERA2 reanalysis. And then Rich just nicely explained these process-oriented water tags, and so now I'm going to show you some results over the US using those tags. We were able to add these to the low resolution run. So we've got the rainout effect tag, evaporative source delta O18 and relative humidity, condensation temperature, as well as mean evaporation source location. Just get my water. Feeling dry. Okay. So using these 2 different tools, I want to address a few different questions. So first, what are the dominant drivers of monthly mean isotopic variation over the US? Where and why do simulated isotopic variations capture the observations? And where does the higher grid resolution fit the data better? Where do we actually see an enhancement with that added higher grid resolution? And so first, before getting into the isotope tracers, I just want to show the benefit that we get in terms of precipitation amount. So in order to do that, I'm comparing the simulation output with the North American regional reanalysis. And both of the nudged simulations capture precipitation patterns fairly well, but where you see the highest error is in the Western US over these, mountainous regions. And that is exactly where you see the highest or the greatest improvement in precipitation amount with that high resolution grid. And so now to attempt to show you kind of the difference that you get in terms of the precipitation delta O18 from this high resolution grid, I'm showing you monthly mean maps over the US of precipitation delta O18 and also the data from US NIP. And so I'm sort of marching through each month just to show you. So first, you can kind of see that the large scale pattern is similar between the low resolution and high resolution grid. But the thing I want you to notice is sort of the much higher amount of variability in both spatial dimension as well as the temporal dimension for precipitation delta 018. And so how does that actually translate into fit with our observations that we have? I'm using two different metrics, mean absolute error as well as R² values. And so in the top, at each US NIP site, I'm just showing you the absolute values for the high resolution case. And so both of the simulations, the high and the low res case, actually feature the same sort of patterns of agreement where you have the best agreement with the observations in the continental interior, and then the worst agreement with the observations along the coastlines. And you don't see a huge change in mean absolute error due to the higher grid resolution. There's not a huge change. You see some improvement in the R² value with high resolution. But this feature is this kind of pattern is the same regardless of resolution. And so in the south, this is where you kind of have the worst agreement, and in ICAM 6, it predicts lower delta 018 of precipitation compared to US NIP, and that's not improved with high resolution. And so just to get kind of a better understanding of why we're seeing this data model fit, we're using our process-oriented water tags. So out of all those tags I listed, these are kind of the three leading factors in terms of determining precipitation delta 018. So you have really high correlation of your rainout fraction. Condensation temperature is very important in the continental interior. And then you've got your delta 018 of your evaporative source as another factor that can be very important in the coastal regions and the south. And so in the center of the continent, where you have relatively high rainout fraction and then larger changes in temperature, that's where the model is doing a relatively better job. However, along those coastal regions, that's where your rainout fraction is relatively lower, your temperature is higher, and so the initial conditions become more important. And so one detail about these simulations that I didn't mention before is that those prescribed sea surface conditions are 1 degree in both simulations. And so we didn't sort of try to enhance the resolution of sea surface temperature or seawater delta 018 in that area of grid refinement for the ocean. And so that could be an opportunity to improve those coastal values in the high resolution case. And so now here's sort of the same plots that I showed you before, but with precipitation d-excess now. So monthly mean maps of precipitation d-excess marching through with the US NIP data. And again, I just want to kind of point out to you between the low resolution and high resolution grids, the large differences in spatial and temporal variability that's captured between these two grid resolutions. And also you can see much more variability in the US NIP data of these d-excess values. And so here's those same sort of metrics, but now with precipitation d-excess, where you have mean absolute error and R² values in the high resolution case on the top, and then the difference between the high resolution case and the low resolution case on the bottom. And so blue indicates where higher grid resolution gives you an enhancement in data model fit. And so in contrast to precipitation delta 018, we see an improvement across almost the entire US with higher grid resolution. And so that's encouraging. And so for our process-oriented water tags, the two kind of leading factors are, the relative humidity of the evaporative source conditions, which makes sense, and also your rainout fraction can become important in sort of some regions of the US. And so a better representation of precipitation d-excess indicates to me that we're improving the representation of those evaporative source conditions. And so let's look at one additional process-oriented tag to see where is that water actually coming from that lands over the US. So the tag in here is sort of showing you the mean location of evaporative source water.
Question.
Do you have a question?
Oh, I'm sorry. It was an accidental press. I'm
Oh.
so sorry.
You're just like, yeah, cool. I like this slide. I hope that's what you meant. Okay, so this is the mean location of evaporative source water. So the tag doesn't give you information about sort of the distribution or extent of different source water regions. It gives you sort of the center of gravity of where your source water is coming from. So these circle markers are each of the US NIP sites, colored according to longitude, and then the diamonds are the mean annual evaporative source water location of the same color. So on the West Coast, you see those warm colors are farther out into the North Pacific. And then as you move to the right or to the east, you see the sort of the mean evaporative source location moves to the east, but also kind of closer to the endpoint of where it actually lands. And so what I kind of want to point out with that is that a lot of those sort of center of gravity locations are falling within that refinement of the grid. And so that's demonstrating to me that including your source locations in your extent of refinement could be really important for precipitation de-excess in your region of interest, because you're improving the evaporative source conditions there. Okay, and then just to kind of try to connect this back to paleo applications with this modeling configuration, I'm trying to kind of take some lessons from this for, future work. So the first thing is that I think these process-oriented tags are really useful for learning about the isotopic controls in your region of interest. And so they are expensive to add to your simulation. So it could be a good idea to add it to a global low resolution simulation to learn about your region of interest before spending lots of resources on a high resolution or variable resolution run. And then in terms of thinking about paleo and the extent of the region you might want to refine or the resolution you might want to capture, there's kind of two things that I'm thinking about. The first is that idea of getting good values of coastal precipitation delta O18 might require thinking about a finer scale variability in your prescribed sea surface temperatures and seawater isotopic composition. Because in this case, we just did 1 degree everywhere, and it could be worth that effort of sort of increasing the resolution of those prescribed boundary conditions in your region of refinement, either using a high resolution simulation that already exists or proxy data. And then what I mentioned before, which is including important evaporative source regions in your region of refinement could really help with precipitation de-excess in your local region of interest. And so then I'll just finish with some conclusions and future directions. So I hope that I've shown that variable resolution CESM is a powerful and cost-effective approach for providing insight into the controlling conditions of observed isotopic variation. We can evaluate isotopic models at fine spatial scales closer to observations with this approach. And also the high resolution grid provides improvements in de-excess over the US and precipitation amount. And then our future plans include applying this configuration to paleoclimate intervals. And that's it.
Okay, do we have questions?
So there are areas where you saw a lot of improvement with high resolution seem to be where you have topography, but these evaporative source regions are over the ocean. So I'm curious what the improvements are there for going higher resolution.
I mean, in terms of the tags we're able to include for, you mean for de-excess?
Yeah, well, for anything, I guess.
Yeah. So in Rich's talk, he mentioned there are like multiple conditions like temperature, wind, and relative humidity of your evaporative source locations that highly predict your precipitation de-excess locally. So my idea is that basically by improving your atmospheric grid resolution over those evaporative source regions, you're getting a better representation of temperature, relative humidity, and winds in that location.
Mm-hmm.
And so that is helping your precipitation de-excess locally at your site.
And it can be pretty fine scale, this variation of the atmospheric conditions.
Yeah.
Okay.
Yeah.
And we have a question from online.
Hi,
yeah.
thank you for the excellent presentation, Sophia. So I have one quick question. It's like, I understand that the motivation for going at monthly scale was for the application for paleoclimate. So my question is whether or not there is any plan to do modern day like simulations at a higher resolution, like higher temporal resolution, I mean, like at maybe weekly or daily or sub-daily timescale.
Oh, yeah.
And another question is that whether or not US NEEP dataset provides observations finer than monthly.
Yeah. So the answer to your first question is that for this presentation, it was really, I was just trying to kind of give a broad scale introduction to the simulation output, but I've got, 6 hourly output and a lot of different variables to look at that I just wasn't able to cover in this presentation. And I'm kind of interested in comparing that weekly scale output of storms with the US NIP output. And so, yeah, the US NIP data set is weekly scale observations. I just, I showed them in terms of monthly means.
Okay, perfect. Thank you so much. I think I will be in touch.
Oh, yeah, sure.
I think we can take another quick question from Clay.
Hi, interesting results. I was just curious if you compared with other variables like just precipitation itself. I'd assume the high resolution would improve the precipitation simulation quite a bit. And so I was curious then if, it's not really improving the delta 18O, but it is improving the precipitation, what that might mean for what resolution is important for paleoclimate simulations. And if, if we're really looking at the delta 18O, how important that may or may not be.
Yeah, I have one slide on that at the beginning where I show just the precipitation amount. I did like a quick comparison on just mean annual precipitation. And so you see a lot of improvement. These are both nudge simulations, by the way. And so you see the greatest improvement in precipitation in those mountainous regions. But an interesting result is that does not directly translate into precipitation delta 018, for instance. So just because you are resolving finer scale terrain in your region of interest does not mean that you will get a better precipitation delta 018. And so what I learned was that I think there are other considerations like the resolution of your ocean condition that matter a lot for your precipitation delta 018 locally. So I'm still kind of thinking about this, but did I answer your question?
Yeah, more or less. I was just curious, like, if how useful it may or may not be for paleoclimate. I think you basically addressed that. Yeah, thanks.
And I do think it would be really interesting to do some, these are just prescribed ocean, but to do some sensitivities with different seawater isotopic compositions to see how that influences the precipitation delta 018 compared to observations.
Great, thanks.
Okay, I think we should move on to the next talk from Jeremy. I will stop sharing here. And by the way, Sophia, David has a question and in the chat. Oh, will you please answer in the chat?
Yeah. Oh, that's right.
Feel free to start your screen sharing, Jeremy.
Yeah, let's see if this works.
Perfect.
Okay, can everyone see that? And everyone can hear me fine?
Yes.
Cool. Well, thanks for organizing this. What I'm going to talk about today is work actually that my former master's student, Ellie Driscoll, did, a sort of data model comparison project looking at greenhouse climates over Eurasia as a way of trying to understand how hydroclimate responds to higher CO2 and using stable isotopes to do that. This is work mostly that Ellie Driscoll led, along with Michael Needham, who's a PhD student with Dave Randall in the Atmospheric Science Department here. And I should preface this, I'm not a climate modeler, I'm more of a data person. So one reason I'm presenting this is also to get feedback on maybe what we might do next, or what things we perhaps even misunderstood about the model that we were using. So to motivate this talk, what I want to show is kind of the uncertainty of hydroclimate projections as CO2 rises. This is the multi-model mean from RCP 8.5 using the C4 MIP models, the end of the century versus end of last century, basically. And in shading is the runoff sensitivity to warming. The stippling is where I believe 6 out of the 8 models agree on the sign of the change. And you can just see that there's large areas of disagreement over the Earth, with potentially places where runoff increases, runoff decreases. Eurasia, which is the area of focus for this talk, is perhaps maybe the least constrained on what runoff will do, or perhaps nothing will change. A lot of this comes down to difficulty in representing the land atmosphere response to CO2 and warming, complicating these hydroclimate projections. So what I want to do is use oxygen isotopes preserved in proxy materials from the Cenozoic to test how hydroclimate changes with warming. Rich and Sophie gave fantastic backgrounds on oxygen isotopes. So I'll just a little bit zoom through how I, as a geologist, tend to think of oxygen isotopes in a very simplified system. So we'll just imagine this is our continent, Eurasia. We have an ocean, and we have the land. Water evaporates from the ocean and gets transported over land, where it progressively rains out over land. Evaporation and transpiration can resupply the atmosphere with moisture. Transpiration resupplies it typically with high delta 18O moisture, evaporation with low delta 18O moisture. This water then gets preserved in proxies. And in this talk, we'll be talking about tooth enamel proxies, lacustrine carbonates, paleosol carbonates, and speleothem carbonates, all authigenic minerals that lock in, or are thought to lock in, water isotopes. If I plot this graphically against longitude across Eurasia, your delta 18O of precipitation decreases as you go across the continent. This is in fact where like the concept of continentality in delta 18O was first enumerated. If I have a low P to ET ratio, that is, I have a lot of ET, primarily T, relative to P, delta 18O will decrease less than it would than if I had a high P to ET ratio. So if I'm raining out a lot relative to the return of moisture to the atmosphere, then I will steepen this gradient quite a bit. Now we can think about how some maybe intervening processes might affect this longitudinal gradient in delta 18O. So let's just pretend, and this has happened in Eurasia before, that we have a big ocean in the middle of the continent, or we have a big mountain range. Well, if we have an ocean, I would expect that ocean to supply the atmosphere with relatively high delta 18O moisture. So as I move across the continent, I might sort of shallow this delta 18O gradient. Similarly, if I go over a mountain range, I force a lot of precipitation relative to ET. There's a lot of runoff. So I might lower or steepen this gradient as I move across the continent. So these are just kind of our first order expectations as we might look across data across an entire continent in the Cenozoic. So why Eurasia? Well, it's the largest continental area on Earth. Much of the interior of Eurasia is dominated by a single air mass, which is the mid-latitude westerlies, which source water in the North Atlantic that precipitates over Europe and gets re-evaporated or transpired back to the atmosphere and transported west. So you have this nice long kind of fetch across the continent where you can understand how rainout processes might occur. We can look at the modern delta 18O of precipitation. So this is just all the data we could gather from, say, waterisotopes.org or the WISER database, or even some studies that aren't included in either. In general, you see high delta 18O water in Western Eurasia, low delta 18O water in Eastern Eurasia. Of course, you have very low here in Southern Tibet, having to do with distillation over the Himalayas. and low values up in the Arctic. If I try and limit this to only areas that I think are receiving dominantly westerly moisture, what I'm going to do is plot this then against longitude. And there's a lot of variability here, but in general, there is a decrease in delta HNO with time. This black line is just a linear regression. And this is, I would say, largely inspired by work that Karita did in the early 2000s, looking at the decrease in delta HNO going across Eurasia in both the winter and the summer. So, okay, we have this system, Eurasia, where delta HNO continuously decreases across the continent. There's some variability here having to do largely with moisture mixing in Eastern Eurasia, largely due to monsoonal systems. But for the most part, delta HNO is high in Europe, low in Eurasia. So why the Cenozoic? The Cenozoic is the last 65 million years of Earth history, and it's seen a large decrease in temperature and in CO2. CO2 fell from maybe 1,500 parts per million in the early Eocene, global mean surface temperatures of 15 degrees greater than today, to pre-industrial values of 280 and a mean surface temperature of about 15 degrees. So we have a large range of greenhouse climates that we can interrogate. The other reason to look at Eurasia is we have an enormous amount of proxy data. So we have a lot of data in Europe, a lot of data in Asia, not a lot, unfortunately, in the middle here. This is about 15,000 samples from 440 sites. All this data is now available for anyone to query in what's called the Patch Lab, which is hosted here at CSU. This also includes all the data from North America up to about 2018 or 2019. So let's look at the data. I'm going to plot all the data, delta HNO, water, reconstructed water against longitude. So look at the Quaternary. There's a shallow decrease in delta HNO across the continent. The black line is just a linear regression, and the gray lines are 95% confidence intervals. In the Pliocene, we see potentially even a reversed gradient, though this might be biased by the fact that we don't have much data from Western Europe in the Pliocene. But when we get into the Miocene, we see much steeper gradients of delta HNO, much steeper decreases in delta HNO across the continent. And this continues into the Oligocene, where we have very steep decreases in delta HNO. So in general, there's a large shift in the shape of this gradient between the Pliocene and the Miocene. And this change in the gradient is actually largely driven by a shift from low delta HNO in Europe in the Plio-Pleistocene to high delta HNO in the Miocene through the Eocene. So what I'm going to do now, this is now the modeling portion, is we're going to use ICSM to just see what the gradient would be in both a pre-industrial run and a 4x CO2 run. We're forcing this with prescribed SSTs and sea ice concentrations. There's prescribed pre-industrial phenology, and we don't have a long spin up, just a 5-year spin up to permit the top 1 meter of the soil delta HNO to equilibrate. Why these values? Well, pre-industrial gives us kind of our cool icehouse end member. 4x gives us perhaps our Eocene or greenhouse end member. So let's look at these modeled isotope gradients across Eurasia. Here again is longitude against delta HNO in ICSM. In red is the 4x and blue is the pre-industrial run. If I sort of normalize this and take the delta in delta HNO across the continent, I can see that the gradient in the 4x model is somewhat shallower than in the pre-industrial model. And this kind of tracks, not perfectly, changes in precipitable water across the continent. So precipitable water is much higher, of course, in the 4x model and doesn't change by as much across the continent as the pre-I control model run. If I look at precipitation and runoff, or P over ET, there's not a huge amount of change. In general, P over ET is lower in the 4x run across the whole of the continent. So the model shows us we have shallower gradients produced under higher CO2, and the models predict a shallower gradient with rising CO2, but the data suggests the opposite. So now I'm going to just speculate for the rest of the talk, and I wish I had an answer for you, except I just have lots of reasons that could explain this, and I'm not quite sure how to rule any of them out currently. So of course the proxies have biases, One of the big ones would be evaporation. There's a lot of lacustrine data in this data set. The red lines here in these graphs on the right are if I remove all lacustrine data. So I'm just going to say all of the lacustrine data is useless. It's all evaporatively overprinted. So I'm going to throw it out. We've tried to screen our samples for evaporation beforehand. Even with that, if I take them all out, we still see the same trend. That is a shift from relatively steep delta 18O gradients across the continent to shallow to non-existent gradients in the Paleopleistocene. So I think our results are kind of robust to changes in evaporation. A bigger problem might be seasonality of proxy formation. So this is now the delta 18O of precipitation across Eurasia in the summer in red, in the winter in blue. And you can see there's both a huge change in the gradient and changes in the absolute values. We don't really have great constraints on when many of these proxies form seasonally. Perhaps Paleozoic carbonates form in the warm and dry periods, though the other proxies in this data set are all not all that well understood. And there could have been perhaps a shift in the carbonate formation timing. So between the Miocene and the Pliocene, we might have had a shift in Europe from, say, summertime values to more winter or annually average formation. And there's some evidence for this in clumped isotope data from Central Europe that suggests big shifts in carbonate formation seasonality in the late Miocene. So of course, if Europe changes from summer to winter and Asia stays the same, that would have the effect of shallowing our gradient without actually any real change in the overall hydroclimate per se. So what about changes in paleogeography? There have, of course, been enormous changes in paleogeography in Eurasia. There was, of course, the Tethys Ocean, which was open 50 million years ago. That progressively closed and left the Paratethys, which progressively drained over the past 15 million years. So, there was this big ocean in the middle of the continent. In general, I would think an ocean would tend to shallow our gradient rather than steepen it. And yet when the ocean is at its greatest extent, we have the steepest delta 18O. So I tend to think that our gradients the ocean here is not playing a big role in modifying our continental gradients in delta 18O. There have been, of course, a lot of topographic changes, a ton of work in Tibet and Asia about what's happened to the plateau or other mountain ranges there. However, in general, these ranges are oriented east-west, and they tend to block southerly moisture rather than impede mid-latitude moisture. So I'm skeptical that topographic changes here are what's responsible for driving the change in our gradient observed with time. Perhaps more likely, and there's been some nice work on this recently, right at the Miocene-Pliocene boundary, there might have been big changes in land surface properties. For example, northern hemisphere ice increased, and there were big changes in vegetation at high latitudes. We went from largely boreal forests to tundra, and there's been work showing that this dramatically reorganizes hydroclimate across Eurasia. So that could be another explanation, which is driving delta 18O changes with time. That's, of course, not in our model simulations because we keep the land surface and the boundary conditions constant.
Sorry for interruption. Please
Yes.
Wrap up. Thank you.
Okay. Yeah, so let me just quickly talk then about the model results. In general, increases in specific humidity should result in shallower delta 18O gradients, but increasing runoff should result in steeper delta 18O gradients. P over ET, and therefore runoff kind of actually P over ET decreases in the 4X simulation, but runoff change appears negligible. And as a consequence, I think it's specific humidity or precipitable water that's driving changes in the gradient in the model. The fact that actually in the observations you see steeper zonal delta 18O gradients perhaps means that runoff increases more in greenhouse climates than what models can simulate. And this perhaps has to do with the timing, changes in the timing of precipitation, whether this is seasonality or storm intensity, or perhaps larger decreases to model conductance than what's represented in our model. So I'll just move to the last slide here. In short, the data shows a steeper zonal delta 18O gradient in greenhouse climates, whereas our model simulations show a shallower one. There's a lot of explanations for this, and I'd be curious to see what people think would be useful to tackle next. So thanks a bunch.
Okay, we have time for questions. Rich, please.
Thanks, Jeremy. That was great. You touched on it at kind of one of your last points there, but I was wondering if you could expand a little bit on impacts from CO2 physiological forcing on some model conductance, because I think that's probably one of the most poorly constrained model parameters, right? So what impacts do you think that could have on your results?
Yeah, absolutely. I mean, I think there's sort of this. My understanding is there's this tug of war, right? You have CO2 fertilization that should fertilize biomass and potentially increase leaf area, and that will increase ecosystem transpiration. At the same time, there's more CO2, so plants can be more water use efficient, and that tends to reduce stomatal conductance. So precisely how that tug of war plays out probably varies by landscape or ecosystem quite a bit, and even by background climate. Like you say, I think it's poorly constrained, and of course in our model we're just using prescribed phenology. So some of these stomatal conductance changes I believe occur, but some not. So this is why I put it as a question mark. Maybe it explains the disagreement in our model simulations. But I think one would want to do a lot more tests. We're fiddling with the land surface parameters to really test that. But I do agree. I think it's a promising avenue where there's potentially something where delta HNO in the past might be useful in giving some insight into how these things change.
Cool, thanks.
More questions?
I have a question.
Yeah, Dave.
Hey, Jeremy, this is Dave Bowden from Caltech. I guess I'm just curious, are you going to run experiments where you maybe alter the continental geometry or play around with the topography just to kind of start to isolate these hypotheses you have of why there's this disagreement?
So I would say I would love to. The problem, of course, on my end is I'm not a modeler. So I need to work with people. But that would be the next thing to do. I think actually my impression is there are these simulations out there where people have played with changes in geography that might be able relatively easily to use in an isotope enabled model. Of course, there's always problems, especially in Eurasia, when it comes to reconstructing topography. Because you go back 50 million years ago and the whole area is just a yelling match of how high the Tibetan Plateau is. But it's still only one area, so there's lots of stuff upwind that would be interesting. No, I think it'd be cool to fiddle with a lot of these parameters or boundary conditions and see if it matters. I think a big one would be just even northern hemisphere vegetation changes and ice sheet or ice extent would be a useful one or a useful place to start.
Cool, thanks.
Thank you. And Jeremy, I want to add a comment that we do have quite a lot of paleoclimate simulations published, available to people, but some of the simulation, they are only accessible by using the NCAR machine. So you can feel free to email me. I think those simulations will be a good start for you to do any further exploration.
Yeah, absolutely. Absolutely. No, it'd be a great project. I just also need to recruit a student to work on this. So thank you guys.
The working group page.
Oh,
yeah.
Did I post that?
Yeah.
And we have a working group web page to summarize available simulations. And we are working slowly on adding new simulations to the web page. And people feel free to check it out and reach out if you have questions.
Cool. Thank you.
Thank you. So we are running 5 minutes late. And why don't we stick with the schedule and come back at 10:40? Thank you, everyone.
Is everybody ready to start again? Yes. All right, great. Okay, we have our first speaker after the break, on to 30 stars. Take it away.
Yeah, so hey, I'm working currently in Propulsion Group, University of Oregon, and I'm here presenting some results on Cretaceous sea surface temperature and temperature gradient, building on Chris' former master student Emily's results. So I'm interested by the Cenomanian-Turonian interval, which is roughly 90 million years ago.
Can you talk up a little for us in the back here?
I will try to do so. So yeah, I'm getting interested in this like 90 million years ago climate, for which a paleo record from ocean core indicate like extremely warm temperature. This is like a panel on the left from O'Brien et al. Proxy synthesis. So they suggest roughly temperature over 40 degrees Celsius in the tropics, like the red curve. But what is probably more striking is that if you look at southern latitude records, the blue curve on the top panel, this is 60 degrees Celsius, mostly oceanographic core, and author would suggest temperature up to 30 degrees Celsius on average for sea surface temperature. If you convert that to temperature gradient, depending on the record you use, you would have gradient of temperature being like 5 to 10 degrees Celsius only from tropic to southern pole mostly, which is way lower than present day gradient, but it's also usually way lower than whatever model can simulate using realistic CO2 reconstruction. This is, for example, a data model comparison for the Cenomanian-Turonian from CESM4 model. I zoomed in on southern high latitudes, and the blue curve is like a model temperature. Temp is like roughly 50 degrees Celsius. The dotted points are measured at site location for the black point, and color points are proxy estimate. This is a bit different from the O'Brien synthesis because Clay Tabor use a different way of converting this initial proxy record to temperature. So a way of trying to improve the data model fit is, of course, use a more recent version of the model that is thought to be like better represented at CO2 that time period temperature. But there is also another way of looking at this problem, and it's like to get back to the data, trying to also figure out whether part of this data model misfit can also be due to biased interpretation of the proxy. And the reason for this is that most of the record, especially at southern high latitudes for the Cretaceous time period, is based on delta 18 carbonate measured on planktonic foramifera. And this proxy, when it's used as paleothermometer, it relies on an estimate of delta 18 of seawater, which is difficult to quantify. The reason, for this reason, people would use average value of -1 per mil for ice-free late Cretaceous world, or they would correct this value for latitudinal factor based on present-day latitudinal distribution, like mean latitudinal distribution of delta 18 seawater. And this has, of course, potential to introduce a lot of biases in temperature estimate we have. As an example, I put this plot from Gaskell et al. Technical note, where they calculated the potential offset of temperature we calculate using either delta 18 seawater from isotope-enabled models or classical way of accounting for delta 18 seawater. And striking thing on this map is first that if you compare the two time periods, the offset is different locally, meaning there is a dependence on boundary condition. And also, if you look at early Eocene results, which is like the closest published map for the Cretaceous, there is this huge offset introduced in southern high latitudes. Another reason to kind of revisit this southern high latitudes paleotemperature record is that when you get back to the original publication that published this southern Atlantic value of 27 to 30 degrees Celsius, authors themselves kind of doubt the validity of the reconstruction. The reason why they got this value is because they measure very depleted delta 18 carbonate, minus 4.5 per mil. They use this like -1 per mil delta 18 of seawater, then end up with this temperature. And they have like very hard time to believe that. And actually all the paper is trying to figure out another way of having this very depleted delta 18 carbonate without accounting such a high temperature. And they come up with this idea that delta 18 seawater might be lower than -1 per mil at the site location at the time. Use like very simple mixing ratio model to try finding mechanisms that could explain that, but kind of never managed to have this mechanism that makes sense to them. And of course, at that time, they didn't have this isotope-enabled model that would enable them to kind of look at delta 18 seawater like heterogeneous pattern, especially. So I kind of did both, trying to improve, to use like more recent model to look at this time period and also get back to the record. So I update existing Cenomanian-Turonian, it's not me, it's like Emily updates Cenomanian-Turonian simulation existing using CESM 1.2 with 3 different CO2 that kind of account for reconstruction for this time period. And then I use the value of temperature and delta height in seawater to get back to the record, relying on the compilation by O'Brien, supplemented by Huber et al. Sites along Australia. Here I have results. So I computed delta height in carbonate using temperature and delta height in seawater from the simulation. Here are the 3 simulations. And looking at that, you can see that delta height of carbonate is very depleted at tropical latitude, which is reflecting the higher temperature. Depletion increase with increasing CO2, which is partly due also to increasing global temperature. And what is also interesting, if you look at southern high latitude, especially on the 2 right maps, is that for similar latitudes, the Indian Ocean and Southern Atlantic have more depleted value than adjacent Pacific Ocean. And all the sites from southern high latitudes come from this more depleted basin. If you look at a quick data model fit a site location for the 3 simulation, can kind of see that, so the best fit is with the 3 times CO2 simulation, if you look at root mean square error, but actually depending on the site, it fits better with the 3 times or 6 times CO2, might partly be explained by the large amplitude of temperature variation over this time period and each site not covering the same interval. 2 locations are always very off from this 1-1 line. This is Central Atlantic site. I won't talk about that in this talk. And also like the sites southern Australia that have like the model really struggled to reproduce delta height in carbonate at this location. And we'll get back to that later. But first, I wanted to try understanding how delta height in seawater could account for this depleted value of delta height in carbonate in the basin where we have the data. So the 3 sites are represented by the dotted, by the circle. 2 black ones are the one where the data model fit for delta height in carbonates is firstly okay. And this like magenta site, Southern Australia, is the one where the fit is very poor. What you can see in the right plot is that delta height in seawater is depleted in the Southern Atlantic and Indian basin from roughly 2 to -9 south of Australia. While it's not like that depleted in Pacific basin. For this simulation, like the Southern Ocean temperature is like roughly 22 degrees Celsius, so way lower than initial proxy estimate. I put as comparison the map for earlier Eocene delta height in seawater from Zhang paper. You can see here that the pattern are very different in Southern Ocean, and Zhang had like very depleted delta height in seawater in the Pacific Ocean, which is kind of reverse pattern than what I have. And it's like, I think, one more reminder that different paleogeography, different climate contexts have very different pattern of delta height in seawater, and we need to account for that. So cool, I managed to have this very depleted delta height in seawater where the site have been drilled, which kind of might explain partly why people would measure depleted delta height in carbonate. No, I need mechanisms to explain that. And to do so, let's look at first the freshwater balance. So this is the 3rd plot, annual average. And what you can see here is that there is a huge amount of runoff coming from southern continents towards Southern Ocean. These kind of freshwater, especially in the Southern Atlantic and Indian Ocean, kind of stay stuck in the basin. This is reflected by the salinity plot, where you have lower than normal marine condition toward even freshwater condition at the surface nearby Australia. And of course, this water like runoff is very depleted in delta 18 because it comes from southern Antarctic continent precipitation. Antarctic is ice-free in this simulation. So now we might wonder why this water is stuck there, while in the Pacific Ocean you can see like there is no like low salinity signal. For example, kind of might be explained by the fact that this water is moved around by oceanic circulation. And so this pattern can be explained by very different Cenomanian oceanic circulation compared to what we know at present day. This is the right plot. So this comes from Ladin et al. 2020 analysis of oceanic circulation in Cenomanian simulation. And what Jean-Baptiste Ladin shows is that because of the very different configuration of paleogeography of the Indian Atlantic Southern Ocean basin. This basin is isolated from the global circulation. The Central Atlantic is almost closed, Lake Passage is almost closed, and you have Indian continent being like in southern position, kind of stuck between Africa and Australia. And because of that, you have almost no export or very few export of water from this basin toward Pacific Ocean, which is shown by the barotropic stream function. You do not have like this very strong circumpolar current, such as present day, that move water eastward. So this explains why the freshwater depleting delta 18 is stuck in the Indian Atlantic basin. And if you compare to Pacific Ocean, in this configuration, there is a deepwater formation in the Southern Pacific with a lot of mixing, which kind of makes this depleted water brought by runoff with whatever is below that is more enriched. And then you create this dipole pattern. So here we provided kind of a mechanisms to explain this pattern of delta 18 seawater that also explain pattern of delta 18 carbonate. But what is of interest here is, of course, temperature. So I did use delta 18 seawater from the model, delta 18 carbonate from the core to compute a new temperature. This is the orange curve. So there is 3 sites here. I applied loss regression to have the curve. You can see that new estimates for temperature are between 18 and roughly like 26 degrees Celsius. On average, most of the points have like -7 degrees Celsius compared to published estimates that are represented by the gray point. And the yellow curve is just for information. This is the only organic record for 60 South, Kerguelen Plateau, so Southern Indian Ocean. And you can see this is like way offset, but I won't talk about that. One thing that you might have noticed is those 5 stars at the bottom left that show, using my methodology, very cool temperature, 5 to 15 degrees Celsius, and it's very hard to believe. CO2 was high, micropaleontological evidence nearby sites suggests there is no cool taxa. Antarctic was covered by forest, very high mean annual temperature. So I try to understand why I have these values here. Of course, we might consider that proxy are not good or biogenetically biased, but there might also be a methodology issue here. So to understand that, I get back to this 25.8 site, which is the very low temperature. And on the right plot, it's like the other side in Southern Atlantic. And I did plot simulated temperature versus simulated delta 18 carbonate for each grid cell from the surface to 150 meters each month. The white color is surface ocean, the blue dark is deeper part. If you look at DSDP 511, the right plot, you can see that there is a very strong correlation between temperature and delta 18 carbonate at that site in the model, meaning that there is no variation of delta 18 seawater seasonally or in depth. But if you get back to this other site, Southern Australia, you can see this very large spread horizontally, especially for location at the surface. And this reflects that delta 18 carbonate is mostly dominated by variation in delta 18 seawater, both seasonally and with depth. And the gray square are where the value of delta 18 carbonate for the proxy measurements. So it kind of fits whatever is simulated for the right side, but for the Southern Ocean side, there is this absence, the model is not able to reproduce delta 18 carbonate from the delta 18 carbonate from the model never match any of the measured data. So either because we have only 5 points, we do not sample the entire water column on season, or just the foraminifera living at that site location just live below the surface. And because I use delta 18 seawater from the surface to derive temperature, I introduce a cold bias. So I also get back to gradient. So I don't show any tropical site results because my new temperature are mostly coherent with whatever was published before, because the model simulates -1 per mil delta 18 seawater, which was the value people use in publication. But if I use the entire, I just temperature to compute gradient, here is the bottom plot. So dotted magenta line is the gradient I have from published data, very dampened gradient. It kind of mimics the gradient computed if you use only TeX86 base pair calibration data. And then if I use my new measurement or my new like temperature estimate, I would have like the two other full lines, so the green and the pink one. So the gradient I calculated are much more higher just because I decrease the temperature from high latitude. Those gradients are closer to simulated gradient that has the dotted line gray. And they're also close to a gradient estimated by Gascal et al. 2022 based on other methodology that rely on comparison compare on the relation between benthic delta 18 measurements and planktonic temperature. One more. Perfect. So I think that that work kind of showed that the peculiar southern ocean configuration due to Cenomanian paleogeography enable having delta 18 seawater that is depleted where the measurement on planktonic foraminifera are done. And this this would result in estimates that are lower than this very extreme temperature, kind of getting back to this Bice et al. 2003 paper. So we have a mechanical proof of what they were suggesting at that time. It results in a gradient being higher, better fit with the model, but there is still discrepancy in temperature. And I think the main out message of this work is that we put a lot of effort comparing data and model in order to improve models. But to be accurate in that work, we also sometimes need to get back to the record, trying to be sure the record is interpreted the right way. And it seems that for the Cenomanian Turanian, at least, it's not always the case. So thank you.
We'll time for one quick question.
Gary Upchurch, CU Boulder. Want to point out that your revised temperature estimates for the Southern Ocean are in pretty good agreement with unpublished and published paleobotanical data, leaf margin data that I calculated for that time interval, around 18 to 20 degrees C, and that makes a whole lot of sense, basically.
Great. Okay. Well, in the interest of time, let's move on to our next speaker. Our next speaker is a remote talk. Oh, here we go. Sorry, we're signing back in right now. Is Michelle there?
I get into the Google Meet.
Yes. Can you hear me?
Yes.
That's the
Are you able to share your screen? Oh, perfect. Okay, great. We can see you in the center mode. So our next speaker is Michelle Fraser. Go ahead and take it away.
Awesome. And you can see it in the correct view?
Yeah, it looks great.
Perfect. Well, hi, everybody. I'm Michelle Fraser. I'm currently a postdoc at Rice University, though this talk is going to be about work that I did as a postdoc at Penn State, working with Lee Kump from Penn State and then Andy Ridgewell from UC Riverside. So my title is Constraining LGM Ocean Carbonate Chemistry and Circulation Using Data Assimilation. So for this project, we broadly sought to explore the role of the ocean carbon cycle in circulation changes in deglaciation in a novel way using data assimilation and an intermediate complexity Earth system model, CGENI. Previous reconstructions of the last glacial maximum compared to the Holocene mostly focus on temperature, but our work uses carbon chemistry proxies to better understand how changes in the ocean carbon cycle affected deglaciation. What started as a bit of an open-ended analysis yielded a particularly interesting result regarding the common narrative of changes in terrestrial carbon storage with deglaciation. For this talk, I will lay out the background of this problem, explain the tools we use to combine data, models, and theory, quickly look at the general output of our reconstruction, consider the key finding, and end with takeaways and ongoing work. And I do want to note here, I've kept this presentation in the shorter AGE-like format, so I'm hoping we'll have a lot of time for questions afterwards. So please have those ready, or comments. All right, so some background. The big question is, why did CO2 increase from the last glacial maximum to the pre-industrial late Holocene. The prevailing view and interpretation from proxies of marine carbon isotope composition is that there was a major transfer of carbon from the ocean to the atmosphere and from the atmosphere to the terrestrial biomass, with a residual increase in the atmosphere. As can be seen in this diagram here, I hope you can see my cursor, from a review paper by Cofield and Ridgewell, there are various proposed components affecting the change in CO2 levels. For example, changes in the ocean circulation, here in the blue, mainly a strengthening of the AMOC, likely release sequestered CO2 in the atmosphere. We can reconstruct delta 13C from benthic foraminifera. For example, a study by Peterson et al. With 480 records used averaging methods to calculate a whole ocean increase in delta 13C of about 0.34 per mil. This work, this by Peterson and the literature consensus, explain this increase by an uptake in carbon by the terrestrial biosphere, right? And you'll see that here as a negative and this change in CO2. And these works have used vegetation models to estimate the loss of carbon in the ocean in the hundreds of petagrams. So, but as you can see in this diagram, this terrestrial uptake of carbon would work to counteract the increase in CO2 we see with deglaciation going from the LGM to modern day. So for my work, our goal was to use data assimilation and the CGE model that I'll introduce in the next slide to constrain changes in ocean circulation, delta 13C, dissolved inorganic carbon, etc., through rigorous reconstruction. So in terms of tools, CGE is a chemistry-enabled EMIC, which includes biogeochemical cycling, ocean circulation, ocean atmosphere and ocean sediment exchanges, as well as the geological cycle of carbon. For this project, we used a 60-member, 20,000-year ensemble for both the LGM and the Holocene scenarios, with new corrections for ocean volume. We created the ensembles by Latin hypercube sampling of 3 tuning parameters that affect ocean circulation: the wind stress scaling, which affects the general circulation, freshwater flux, which affects North Atlantic deep water formation, and brine water rejection, which affects the Southern Ocean source. I imagine this is going to be familiar for a lot of you, but for those who aren't familiar with data assimilation, bear with me for this slide. In model studies, proxy records, we often use them to assess the validity of our simulations. Data assimilation is a statistical method to do that, to merge observations and model predictions to infer system states and parameters. So instead of just comparing the two, we use both the proxy data and the models to basically merge them together into a new, better reconstruction. So here you can see the defining equation, which is solving for the analysis or posterior, that's X sub A there. And the posterior is this updated ensemble such that the climate states account for robust information from the proxy records. It's a function of the background or the prior, which is the climate model ensemble. The innovation is the difference between the data from the proxy records and an estimate of the proxy data from the climate model output, generally using a proxy system model. The innovation is weighted by a Kalman-Gain matrix, which accounts for uncertainties and covariance. So our innovation is this benthic delta 13C record from Peterson et al. And since CGE directly calculates delta 13, we don't have to use a proxy system model except to check our results for robustness. And as a further measure of robustness, we experimented with all kinds of values for data uncertainty, localization, regridding options. These choices were made really easy to implement in our data assimilation framework because of the Dash MATLAB toolbox, which is developed by Jonathan King and is available on GitHub. All right, so let's look at a couple of plots here of the reconstruction. So these plots represent some of the results from the reconstructed ensemble mean for both LGM, which is on top, these 2 plots, and Holocene, which is here on the bottom. So on the left, we're looking at benthic delta 13C. The little colored circles are showing the proxy data sites and values. You can see it's heavy, the data is heavy in the Atlantic. In both cases, the Atlantic has higher values than the Pacific, but the stronger signal is higher values in the Holocene, right? So we're seeing higher values of delta 13C here in the Holocene. So one of the advantages of data assimilation that you can imagine from this plot is the ability to fill in areas of sparse data coverage, like the Pacific, with robust model relationships. So instead of just doing some sort of weighted average of those few points in the Pacific, we can think how the model would relate what's going on in the Atlantic to what's going on in the Pacific to better correct for those values. On the right, I'm looking at slices of the Atlantic zonal average values for delta 13C. As expected, the LGM is showing a much stronger vertical gradient. And here you can see really good matching up with the proxy data, which is encouraging. I'm not going to show you a bunch of other plots. I want to highlight just for this talk one key finding. There's a lot of numbers here, but I'll explain them. When we look at these high-level carbon inventories from the data assimilation, we can see an increase in delta 13 from LGM to Holocene as expected, right? So here, the LGM DA, our ocean delta 13C goes up from 0.23 to 0.45. So that's a whole ocean increase of about 0.22. Pretty similar to other studies. And we've got error that we've assessed from our ensemble spread. But what's interesting about our results is that this increase in delta 13C is accompanied by a small, insignificant decrease in DIC, the dissolved inorganic carbon, right? So it's going down, ever so slightly. It's basically identical between the LGM and the Holocene, right? So we've got our expected change in delta 13C, but it's accompanied by an insignificant DIC change. And so the question is, what does that mean? Keeping in mind, we were expecting in terms of other studies that you're going to see this huge increase because of this uptake by the biosphere as you're losing the ice sheet, right? So it means that from the LGM to the Holocene, we're seeing an increase in isotopically heavy carbon in the ocean and the atmosphere without an overall increase in total carbon. And so this is where we can start postulating what might be going on, right? We could be accumulating isotopically neutral carbon, for example, from river input, while burying marine organic matter, which is isotopically light. It could be that the terrestrial carbon growth is pulling out light carbon, but will also accompanied by some sort of large increase in neutral carbon, right? So remember that if the delta 13C change was only by terrestrial carbon change, there'd be significantly less DIC in the Holocene. So the conclusion from these results is that one cannot conclusively interpret this ocean delta 13C change in terms of terrestrial biomass growth. So as you might imagine, one of our next steps is to try to understand if it's not from terrestrial biomass growth, what is going on? Why is there this ocean delta 13C change? I really hope to have new results since AGU, but I've actually been on medical leave. So I'm open to people's predictions and thoughts on what might be happening, but a couple of ways we're going to be exploring it. One of the things we're doing is we'll be adding the sediments module to CGenie to also assimilate not just the delta 13C, but also calcium carbonate. This will allow us to add whole ocean alkalinity as one of those tuning parameters in our Latin hypercube sampling. And then this is a fun test because if the parameter that's sort of selected by the DA is similar to modern alkalinity, that can tell us we're doing something right. And we're curious to see if we'll see the same carbon storage results when adding a second assimilation factor. But then we also want to know, where are these delta 13C changes coming from, if not from the terrestrial carbon storage, right? So we can add tracers to explore the carbon cycle, which is the next step that I'm working on at the moment. Right. So the primary takeaway from this talk is that with a range of data assimilation solutions, our results are contrary to the conventional interpretation of an increase in delta 13C in the Holocene, corresponding to uptake by the terrestrial biosphere. We also demonstrate the power of combining models with proxy data through rigorous data assimilation methods. So it looks like we have some time for questions. I look forward to discussion, and if you want to discuss further, there's my email there as well. Thank you.
Okay, questions?
I have one.
Here. Okay, yeah, we're not updated.
Okay. Hi, this is Vince Cooper at the University of Washington. Excited to see the application of data assimilation. I'm curious if you could tell us a little bit more about where spread in the prior comes from. Like, are you changing parameters in CGenie, or how do you have some kind of spread to start off with in the prior?
Right. So I went through this quickly. There's 3 parameters that we're changing in CGenie. These parameters are used to tune the ocean circulation CGenie. So one is basically controls the strength of the AMOC through like brine water rejection. There's these 3 different circulation tuning parameters. And you're going to expect that you're going to get a different configuration in those between the LGM and the Holocene. But what we do is we have this 60-member ensemble. We ran the same 3 parameters for both. Obviously, a lot of other things are different between the LGM and the Holocene, continental configurations and things like that. But we use Latin hypercube sampling of these 3 parameters to come up with that huge spread in the prior.
Okay, that makes sense. Thanks.
And what we're hoping to do when we add the calcium carbonate is add a 4th parameter that's a tuning parameter, which is whole ocean alkalinity.
Cool. Thank you.
Yes.
Hi, Michelle. Nice talk.
Hi.
I have a related question about the baseline climate in CGenie. I'm wondering whether it is worth exploring the sensitivity to the climate, for example, the surface wind, temperature, etc.
Right, the climate sensitivity. I mean, I know that's been done by a previous postdoc who was working on similar work at Penn State, who was doing that for the PETM, and he worked on changing the climate sensitivity. I'm not sure that's necessary when we're dealing with this. And we're not using a temperature proxy either. We're not constraining the ocean temperature. We're really just constraining the DIC, the delta 13C, and then hopefully calcium carbonate. We're really focused on ocean circulation and less about, surface temperature, atmospheric temperature. But yeah, I mean, that is an interesting question. Do you have any suggestions or thoughts on that? Because you know more about this than I do.
How about surface winds? Will that make a difference?
Right, so they're being forced with a climatological value, I believe, but it's different. I got to remember because Andy helped me set these runs up, obviously, the 2 different configurations. That's one of the things that's going to look different between the LGM and the Holocene. But one of those tuning parameters we're varying is basically the coupling between the surface winds and what's going on in the ocean. So that allows us to very freely match the proxy data and allow for different ocean circulations between the 2.
Okay, thank you.
Yes, go ahead, Keith.
Hi, thank you for that interesting talk. It's one thing that wasn't clear to me is with adding alkalinity to your system, are you going to be prescribing the inventory of alkalinity, or are you solving for it? Is there sufficient observational constraints on the ocean's inventory of alkalinity in the LGM?
Right, so the whole ocean alkalinity is basically a tuning parameter within the model. And no, we don't have sufficient observations in the LGM. So that's like something that we'll be solving for in a sense. That's something we're allowing to vary and letting the DA pick the best thing. But we do have a sufficient constraint on the Holocene. So if the DA, the data assimilation, sort of picks a crazy value of whole ocean alkalinity for the Holocene that doesn't match observations, we know that something's going wrong. So that'll be like a nice sanity check. I don't know if that answered your question.
So I'm a bit confused. I'm hearing you say that there's not sufficient obs to constrain it, but it's a free parameter that you're going to be solving for. How that sounds like it's going to be an underdetermined equation you're trying to constrain then.
Yeah, so what we're constraining is the calcium carbonate in the sediments. That's what the proxy data we have.
Okay.
And so to create that spread in the prior, we're going to be feeding in different values of whole ocean alkalinity, as well as the different values of the different tuning parameters related to the circulation.
Okay, so would you start with like a prior of LGM inventory is the same, but the concentrations are different because of sea level rise or something like sea level change?
Well, the LGM versus the Holocene, those two different configurations, we have different sea level rise, different ocean volume, different concentrations. Like we've already done of a bunch of different variables. So we've already done that. Just we already have the two different configurations going on.
with alkalinity already.
Right. So alkalinity, we're gonna expect that it'll tune and pick different values. So for example, in one of those ocean circulation parameters we're varying is basically controlling the strength of the AMOC, right? And if we use data assimilation on the LGM and the Holocene scenarios, it's going to pick out totally different values for that AMOC parameter, right? It should be, because we're working with different delta 13C values for LGM and Holocene, it should be figuring out that we had a weaker AMOC in the LGM, but we're not constraining it on that. So it's going to be the same thing with alkalinity. It should figure out that the alkalinity is different than it was in the Holocene. Does that kind of make sense?
I think so.
Okay, sorry. When it comes to the carbon chemistry is like new to me. I switched to this field from cloud physics. So I'm still learning, but I wanted to learn data assimilation, get involved in paleoclimate.
Okay, thanks, Michelle.
We have time for one more question or comment.
Good. I'm from Drew, postdoc at UNCAR. I'm just wondering how reliable is the spatial structure resolved by Gini, because that's essentially you are relying on to propagate pricing information to locations without observations.
Right. So, I mean, there's two questions there. There's like the vertical structure and then the horizontal structure. Because one of the things we actually varied in sort of coming up with like a robustness is what you do with proxy data points that are like above or below the model grid. All that to say, I feel good about it from a vertical gradient sense because we saw robust results even when we varied some of those assumptions. The concern of lack of data in the Pacific is very concerning, but we believe that the data assimilation proves a better method than either just running models or just doing some sort of spatial weighted average of those few variables, right? Because we can say now if the model is always showing, and I'm just going to be making up these relationships, the model's always showing higher D13C in the Atlantic than the Pacific, then that helps to sort of validate if we're seeing that signal in the prox data as well. That's sort of the beauty of data assimilation, is it combines the two. But yeah, I mean, obviously, a next step for people who actually do this sort of thing would be to collect more data in the Pacific.
Thank you.
Great, thank you, Michelle.
Thanks.
In the interest of time, we're going to move on to our next speaker, remote speaker. Kirsten, are you online?
Yes, I am.
Great. Are you able to share your screen?
Yes. One moment, sorry.
Yeah, no problem.
Where did it go? Oh. Sorry, I'm having a privacy issue.
We have your slides here in the room. You want us to share the slides for you? That's also possible.
Sure, that's easier.
See if we can. Okay. Do you want to do it? Okay. Okay, great. Can everyone online see these slides? Okay, great. Thanks for the thumbs up. Okay, our next speaker is Kirsten Kopnick. Take it away.
Hi, everybody. I'm Kirsten. I am a graduate student working with Ellie Zipperman at Harvard University. And this project is in collaboration with Dr. Minmin Fu at Yale. And we're looking at how dominant is surface mass balance of the Laurentide ice sheet during the last deglaciation, and we're using iTrace. Do I just say next slide? Okay, so a little bit of motivation behind this project is we can look at the path or the ice volume history of the last deglaciation through glacial isostatic adjustment and sea level indicators. But the primary role of climatic feedbacks that might aid in this evolution are unknown. Next. And so here we have plotted the ice volume height as a function of time here with each time slice of ice 6G. And so you can see that as time evolves, that we are lessening in ice through this ice path. Next, please. So I guess a motivating question and our goal for this project is kind of to look at how the atmosphere and the ocean might interact with the ice sheet to determine accumulation, ablation patterns, to look at how orbital configurations play in the role. Are they the determining factor? Is it perhaps CO2 or meltwater forcing? Next. So in this work, we are attempting to assess these climatic feedbacks using a surface mass balance parameterization of the Laurentide ice sheet using iTrace. Next. So what is iTrace? iTrace is a transient climate simulation of the last deglaciation from 20,000 years ago to 11, and it's performed with the isotope-enabled version of CESM 1.3. Next. So it uses prescribed ice sheet boundary conditions from ice 6G and has a non-active ice sheet. Next. Yeah, thank you. And so it uses 3 different forcing parameters with CO2 or greenhouse gases, the insulation, as well as meltwater fluxes or forcings. Next. And so it's broken into 4 different experiments. So we have the ice experiment, which is just evolving ice sheets and ocean bathymetry and holding all other forcings at LGM conditions. Then we have the next experiment, which is ice and ice plus orb, which is evolving both ice sheets and orbital conditions. Next, we evolve, or they evolve ice sheet, orbital conditions, and greenhouse gas concentrations. And finally, the fully forced experiment, which has evolving ice sheets, orbital conditions, greenhouse gases, and meltwater forcing. Next. So the way that we look at this is through a surface mass balance. And so first we use a kind of melt parameterization to compute the annual melt. Next. So it's just a sum of the, it's just an energy balance. And so next. So here we have our shortwave radiation, our net long wave, sensible latent heat flux, and then of course geothermal heat flux. And since it's a energy balance, we divide by the latent heat of freezing. Next, sorry. Latent heat of freezing and density of water to convert to meters per year water equivalent. Next. Through a sign convention, just a note here. And we will actually take the numerator to be just the FGR component from the land model. So next. One more, yes. So as we know, the kind of top layer of an ice sheet is a semi-permeable layer that allows for the refreeze of part of meltwater. And so to compute this fractional refreeze, we follow this paper, Colonial et al. In 2009, next, which is just a function of snow accumulation and this melt that I just talked about. So this is based off of a suite of papers, but does a nice job of estimating the fractional refreeze. Next. So then surface mass balance annually is given by thank you, snow plus snow accumulation, PS, plus the fractional refreeze of rain minus our sublimation evaporation, and then that fractional refreeze of melt. Next. So one note is we are assuming that surface snow redistribution through snow blowing is very small. So this difference between So we are taking the difference between surface mass balance as well as the total ice volume change to be attributed to ice flow, calving, and other ice sheet dynamics. Next. So I want to say that these are incredibly preliminary results as of probably last week. So take this with a grain of salt. But here I am showing the surface mass balance compared to ice volume change of I6G, which is given in the black line. And these colored lines here are the surface mass balance, the annual surface mass balance of each experiment, where the purple line, which is what we'll focus on for this talk, is the fully forced iTrace experiment. The shaded region here is actually the annual surface mass balance, and the thicker line is the smoothed version using a 100-year window and a linear fit. So you can see that there's actually a lot of variability in the surface mass balance as well. And so one thing to note is that the surface mass balance here is actually positive. So this actually indicates growth, which is probably not very conducive towards undeglaciated climate. So next, please. So here we have this really massive increase in surface mass balance variability between 12 and 11.4 Ka. Next, please. So all these experiments with this parameterization drastically underestimate surface mass balance and actually predict a positive growth of the ice sheet. So it raises a few questions for us so far. Is our assumption that melt is the heat flux divided by latent heat of freezing, et cetera? Is that correct? Are we missing perhaps a factor here, which I have a guess towards? Or does CESM predict too much snow accumulation? What exactly happened at 12 Ka between 12 and 11.4? Is this the Younger Dryas? What is going on there? Next, please. So we can look at the spatial distribution of the surface mass balance as well. Next, thank you. And we can look at the surface mass balance of each grid cell across the Laurentide ice sheet. And then we average this just over the last over 100-year period centered at each of the time slices. Next. So we see this orographically induced precipitation along the western side of the ice sheet from the moving of the jet stream. Next. And we see this marginal melting area. However, we see that, as we saw earlier, that maybe the magnitude of the melting is not quite accurate, or who knows. Next, please. So again, we see too much accumulation and maybe not enough melting, and the spatial distribution of melt kind of looks right, but maybe not the right magnitude. Next, please. So then we can look at the integrated surface mass balance and compare it to the total ice volume of I6G over time. Thank you. And so in red is the ice volume over time of I6G, and in the colored lines, again, are our experiments integrated over time in those 1,000-year segments. And so we can see that clearly something that we're showing this growth again by the upward trajectory of each of those time slices. And the stair-stepped pattern is just because these runs are given at 1,000-year increments. So we're kind of reinitializing each run every 1,000 years with a new ice topography. Next, please. So again, the overall growth, we see an overall growth of ice, and that there's not this ice sheet decay as we would expect during this deglaciation period. There's also kind of like a slight decrease in the rate after 14,000 years ago. Next, please. So then we're going to look at the increased variability between 12 and 11.4. And so here in this top panel, we show a zoomed in version of the yearly surface mass balance of iTrace, the fully forced experiment. And we see that dramatic increase here. And then in that bottom left panel, we are looking at the net melt here, and we see a corresponding increase in variability. And then in the bottom right panel, we see a slight increase in variability during this period in the snow accumulation. So next, please. So it looks like net melt contributes the most to this increase in variability, although there is a slight increase in variability in snow accumulation. Next, please. Next again. And so if we take the standard deviation of the 50 years before the transition, so during the period of the normal variability, and compare it to the 50 years after, and look at the surface mass balance, the net melt, and the snow accumulation. Next, please. We can see clearly here where the leftmost panel is the surface mass balance standard deviation difference is very similar to that of the net melt, which is the middle panel here. So next, please. So from this, we can clearly see that the variability in the net melt seems to contribute the most to the increase in variability in annual surface mass balance, although there is a slight increase in variability in the snow accumulation. Next, please. So you can just put all the until the, thank you. So the summary here is we used a surface mass balance parameterization that includes a refreeze term to study the ice sheet dynamics of the glaciation, specifically of the Laurentide ice sheet, using this transient simulation iTrace. And so we found so far that surface mass balance over predicts ice volume growth for all of the experiment, and there might be a slow decrease in the growth rate after 14, but predicts a positive surface mass balance. We also see an increased variability in annual surface mass balance in the fully forced experiment between 12 and 11.4 Ka due to the increase in variability in net melt. So some future work, you can just go to the, yeah, there we go. Thank you. Is we'd like to investigate if there's a better way of estimating total melt. Yeah, so where, and then also additionally, where is this variability at 12K coming from? And then finally, in addition, or maybe supplementary, we are going to run our own CESM2 experiments with different ice topographies and other initial conditions in order to examine this relationship in more depth. So although one thing is to note is if these results still hold true, let's say next week, then and if surface mass balance is truly not a great indicator of deglaciation or glaciation in this case, then these experiments might need to be done with an active ice sheet. And with that, I'll say thank you and open up to the floor to questions. And thank you to whomever was clicking for me.
Great, we have time for questions.
Maybe I can ask one question. Hi, this is John. I'm wondering about maybe you showed that and I somehow missed. What's like the river runoff and the ice runoff in your calculation? Has that been accounted for?
I didn't quite hear you. I'm so sorry. The ice runoff?
Yeah.
Yeah, so it is not being directly accounted for unless, so we're just assuming that we get some sort of total melt from the surface energy balance and that it's either being refrozen into the firn layer or it is just a runoff. If that answers your question.
Yeah, my question is about the runoff calculation from the model output. Sorry, I don't have like experience on this. I'm wondering whether this needs to be accounted for. For example, in the land model, there's some threshold regarding the snow depths. Could be like a 10 or 2 meter.
It's 10 meters.
Yeah, it could be.
I don't know what it was in ICS 1.3. It might have been 2 meters.
Thanks. Could be 10 meter. And if we have snow accumulation, say above the ice sheets, and the height is above 10 meters, then those excessive snowfall will be converted into snow or ice runoff automatically. So I imagine this may impact the overall budget of the mass balance.
Yeah, that's a good point. I'll have to look into that. We're not currently and have that incorporated.
Thanks.
Can I just add one thing there for her? I don't know what iTrace did, but Trace opened up the Bering Strait, but a little bit earlier than 12.9 thousand years ago. So you might just look at what they did with the Bering Strait, because that could have an impact on things like the EMOC and that. I would guess they opened it at 12.9, but I don't know.
Okay, I'll look at that. Thank you.
This is Sophia. I have a quick question because you mentioned on one slide that you suspect there may be a term missing from your parameterization, but I didn't, maybe I missed when you elaborated on that. So I'm curious just to hear what you think might be missing.
Yeah. Yeah, if you don't mind going back to when I'm talking about the melts, that's fairly early on. The melt parameterization. Yeah, there you go. So you notice that I'm dividing this entire equation by the latent heat of freezing to convert from meters per year, or convert from watts per meter squared to meters per year water equivalent. I suspect that maybe that the latent heat flux might need to be multiplied by the latent heat of sublimation or evaporation maybe instead, and then divide everything by that factor, but I'm not quite, I think that I might be not accounting for something quite in this melt parameterization, because I don't think that having that positive of a surface mass balance makes physical sense, in my opinion, at this moment, as of getting these results a week ago.
Thank you.
Yeah.
Unless we have one more quick question. Just want to make sure I didn't miss anyone. Okay, great. In the interest of time, we'll move on to our next speaker. Thank you, Kirsten. Okay, our last speaker of this session is in person. We're just pulling up the presentation now. Okay, great. Our next speaker is Na Wang. Take it away.
Hello, everyone. My name is Na, and I am a postdoc from Rice University. And today I'm going to talk about the ENSO-monsoon teleconnection during the last millennium. And this work is the application of using data assimilation techniques to advance our understanding about the monsoon dynamics. So first, the motivation is that the Asian sun monsoon has a profound socio-economic impacts, and the variations of monsoon rainfall can lead to severe flood and drought. So the monsoon prediction is very important. And on the interannual scale, ENSO is a dominant control on the monsoon variability. And here I show an example in 2015, which is a typical El Nino year. And we can see that the warm SSTs in the tropical Eastern Pacific can associated with the precipitation decreased in India, Southeast Asia, and Northern China. But however, such a relationship is not always reliable, as we can see at the end of the 20th century. The correlation between monsoon rainfall and Nino index becomes insignificant, indicating the failure of ENSO-monsoon teleconnection. So to better constrain the future prediction, we need to know how did the teleconnection change in the past. And previous studies have listed many possible factors, such as
anthropogenic warming and nature external forces such as solar irradiation. And here in our study, we are focusing on the role of internal climate variability. And specifically, we are focusing on the role of Pacific decadal oscillation and Atlantic multi-decadal oscillation, and how they may interact with the ENSO-monsoon teleconnection. So in the context of last millennium, we ask 3 research questions. And first, how do the PDO and AMO in combination with ENSO influence the Asian sun monsoon variability? And second, how do the PDO and AMO influence the ENSO monsoon relationship as measured by the correlation strength? And finally, can we improve the monsoon prediction using insights from the last millennium? We use 2 data assimilation products in our study. And the first is the last millennium reanalysis LMR, which simulates the temperature sensitive proxies with the CESM4 last millennium simulation. And the second is Paleoclimate hydrodynamics data assimilation products, FIDA, which is based on a network of hydroclimate sensitive proxies and use the CESM LME as a multiplier. So the advantage of this product is that they can generate the global gridded data of multiple variables at the same time, and they are also constrained by both physical laws in the model and also the time variations from the proxy records. And beyond that, we also use the full forcing experiments from the CESM LME as a supplement to compare with the data assimilation products. So before jumping to the results part, there is a fundamental question. So can we trust the data assimilation products in our study? And the quick answer is yes. We have put them into text, and we found that they have successfully reproduced the spatial pattern of the teleconnection pattern as shown on the left, and also they successfully reproduced the time variation of PDO and AMO index. And the correlation of the spatial correlation and the temporal correlation are all significant, at least at the 5% confidence level. And to answer the first question, I first show the individual influence of each climate mode on the Asian sun monsoon. So in this figure, the color shading represents the PDSI difference between the warm and cold phase of each climate mode. And the PDSI is a drought index. We use it as a measure for monsoon rainfall intensity. And on the first on the top is the results from LMR, and at the bottom is the result from FIDA. And we found that they both show that the warm phase of ENSO and PDO are associated with overall drier conditions over the monsoon regions. And by contrast, the influence of AMO is more, and the significant signals showing by the stippling area only appear in Southeast Asia. And here are the results from the CESM. We use the precipitation outputs from the model, and these figures show the ensemble mean of the precipitation difference between the warm and cold phase of each climate mode. And we found that the spatial pattern is very similar to the data assimilation products. They both show that the warm phase of ENSO PDO associated with overall dry conditions. So from the individual inference, we can simply expect that when the El Nino occurred on the warm phase of PDO, we can expect even drier conditions in the monsoon regions. But compared to the direction of change, I think what also matters is that the compounding effects is significantly different from the individual influence of ENSO. So to test that, we compared the PDSI difference that responds to El Nino events that occurred under the certain phase of PDO AMO to all the corresponding El Nino events throughout the last millennium. And we found that, so maybe it's not very clear. So I want you to focus on the stippling areas in these maps. They appear in Southeast Asia and India. And this is where the compounding effects are significantly different from the influence of ENSO alone. And we also did the same analysis for AMO, but we did not find any significant signals. And this is not surprising because the individual influence of AMO is more, as shown in previous slides. And here shows the results from CESM. And this is just an example of the combination of El Nino with the warm phase of PDO. But basically, we did not find any significant distinguish between the compounding effects and the individual effects of ENSO alone. So for the first question, we care about the monsoon variability itself. And for the second question, we shift to the co-variability between ENSO and monsoon. So here we use the 20-year sliding correlation as a measure of the changing ENSO-monsoon relationship. And you can see that the correlation coefficients change a lot over the past millennium, and both for the East Asian submonsoon region and the Indian submonsoon region. So our next step is to divide these correlations into different groups. And here are the probability density function of different groups of correlations. And the blue color represents the cold phase of PDO on the left and AMO on the right. And the red color represents the corresponding warm phases. And we found that the blue and the red curves looks very similar. And we did a student t-test for the mean correlations between the warming groups and the cooling groups, the warm phase group and cold phase groups. And we did not find any significant difference. And we also get very similar results from the CESM. The PDFs of warm and cold phases are all looks very similar. So to look at it further, we did another sliding correlation analysis, but use a larger window size of 101 years. Try to connect the changes in PDO and AMO index to the changes of the ENSO-monsoon relationship. And the blue and the red colors in these figures just showing that the influence of PDO and AMO on the ENSO-monsoon relationship is also non-stationary over the last millennium. So if you could see the signals at the bottom of each panel, it just represents the central year of the sliding window, during which period the correlation between PDO and AMO index and the ENSO-monsoon relationship is significant. And you can see they are just appear in a very limited period of the last millennium. And that's why when we consider all the warm events and cold events during the last millennium, we cannot see any significant differences. And finally, in the prediction perspective, we have constructed different linear regression models. And here shows the adjusted R-square for the linear regression model. And basically, the higher the value, the better the model. And there are some discrepancies for the East Asian submonsoon region. But if we look at the Indian submonsoon region, we find that the both data sets show that if we add PDO as an additional predictor, in addition to Nino 3-4 index, we get better prediction for the Indian submonsoon. But if we include AMO, we will see that it makes little differences. And here is the results from the CESM. And the model also shows that the including of PDO can improve the adjusted R-square, but the inclusion of AMO makes little changes. So finally, by integrating all the results from different data sets, we have the following home messages. First, the PDO impacts Asian submonsoon variability more than the AMO.
and its consideration can yield improved Indian sun monsoon prediction. And this conclusion is basically consistent with previous studies, but we further test them in a larger sample size from the last millennium. And the second is that the influence of PDO and AMO on the ENSO-monsoon relationship is non-stationary across the last millennium, which means we must be very careful about the lesson or knowledge we learn from the instrumental period, because they may not apply to long-term changes. And finally, the paleo data assimilation products generate very similar results, like in terms of the teleconnection pattern as in the model. So I think one possibility is that at the current stage, the constraints from the proxy data to the teleconnection pattern is very weak because we have very limited proxies in the Asian region. So in the future, I think it's worth to check what if we increase the coverage of proxy data, will that lead to a different story? And if you're interested in this work, you can check out our recently published paper on CGR atmosphere, and I'm happy for any questions. Thank you.
Okay, go ahead, Mohammed.
Hi, thank you for the excellent presentation. So I have a quick question. It's like, so you use PDO and AMO, but you did not use the Indian Ocean Dipole. Is there a reason for that, or could that influence your presentation or your results? Thank you.
Thank you. Yes. So the reason we use PDO and AMO because they are decadal scale variability, and we found that the ENSO monsoon relationship they changes on a decadal on the decadal scale. But yes, there may be other decadal scale variability in the Indian Ocean. Maybe they also have potentially have important impacts on such a relationship. And I think yes, maybe we can check in the future.
Okay, makes sense. Thank you.
Thank you.
I have a question.
Yes.
You like the non-stationarity was really cool to see. Do you know what gives rise to the reason why you'd have a positive correlation and then a negative? Is it maybe on Mohammed's question? Is it the Indian Ocean dipole, or do you know what mechanism might be giving rise to that non-stationarity?
Now I don't have the answer for your question, but it's worth to check why at some period they have impacts, why in other periods they do not have impacts. So maybe we need to do more numerical experiments to check that. So if we have some force in the Indian Ocean to see if the PDO can have a influence to the teleconnection. Yes.
That'd be really cool.
Yeah.
So the time series plot you made at the very beginning of that correlation becoming insignificant was pretty striking. Has that been detrended? Like the trends in long-term trends in say the Asian summer monsoon? Because I wonder if you're capturing say the anthropogenic effects of aerosols or just global warming versus the role of ENSO or other forms of variability.
I think this kind of, you mean right? This
this one.
It has mentioned in many papers published. I think global warming may be a potential factor, but if we look at the long-term changes, not only the instrumental period, we can also find that the decadal changes of such a relationship. So anthropogenic warming is a potential factor, but we think that there are many other possible factors to influence such a relationship. Yes.
Now, thanks for the nice talk. So I have two comments. So the first regarding the non-stationarity. So it shows that the paleo data assimilation product shows very good skill during the instrumental era. But when you go back in time, the issue of data attrition becomes increasingly worse. So the skill may not maintained very well when you go back in time, especially for hydroclimate. So the second comment is about the space, the data connection scale you see when you do the model data comparison. Because when those two PDA products are based on estimation with CCSM4 and the CSM last Millennium Ensemble already, and they are relying on those spatial teleconnection. So I think it might not be surprising to see very good agreement in that teleconnection when compared to CSM again.
Yes. Thank you.
John?
Yeah, go back to this plot. I think it's very interesting. I'm not following any of the literature here. I'm wondering whether their study focusing on potential role of aerosol forcing here.
I don't see any paper. Maybe there are some paper, but I have not seen any talk about the aerosol role in such changing relationship. Yes.
Okay. All right, if we don't have any more questions, thank you so much, Na. Thank you. Now we're going to break for lunch for 1 hour and return at 1 p.m. Mountain Time. Thank you everybody for attending the morning session, and please return for the afternoon session.